Assalamualaikum students. Before going to our educational topic, please subscribe my channel Muntazir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic. In this tutorial video, we will study about the beta oxidation of fatty acids. Students, beta oxidation is a catabolic pathway of fats in which free fatty acids are converted into acetyl coenzyme A. As this mechanism include oxygen in one form or another, so this mechanism is termed as oxidation. But why is this process known as beta oxidation? The answer is that the fats are stored in the living organisms in different configurations, and one of the major fats formed are the triglycerides. These triglycerides consists of a single glycerol molecule that is attached to three fatty acid molecules. Students, it is very important to understand the structure of the fatty acids. Here are two diagrams which explains the chemical structure of fatty acids. The chemical structure of fatty acids has a long chain of carbon and hydrogen atoms which is denoted as R. This R is a side chain that is associated to the functional group, the carboxyl group, that is showed by COOH. As we know that the side chain R comprises of a single chain of carbon and hydrogen atoms. Let's enlarge this side chain a little. Students, the carbon atom which is directly attached to the functional group is called as alpha carbon, and the next carbon from the alpha carbon is called the beta carbon. Students, during beta oxidation of fatty acids, this side chain R is degenerated between the alpha and the beta carbon, and in this way it takes the name of beta oxidation. Now we consider general details about beta oxidation. For example, the human body comprises of adipose tissues which consists of triglycerides. These triglycerides possess fatty acids and glycerol molecules. These fatty acids are apportioned to all over the human body, and from there they move into the bloodstream. Through the blood, they are circulated to virtually all the cells of the body that can metabolize these fatty acids. Then these fatty acids penetrate inside the cytoplasm of the target cells. During the next stage, these fatty acids pass into the matrix of the mitochondria after passing the inner and outer mitochondrial membranes. During the final stage, the fatty acids are metabolized oxidatively inside the mitochondrial matrix. So the complete beta oxidation process can be divided into three stages. The first stage is showed, that is transfer of fatty acids from the adipose tissues to the target cells. Second stage is showed by B point, that is entry of the fatty acids into the cytoplasm and then into the mitochondria of the target cells. And understand oxidative catabolism inside the mitochondrial matrix. Now we understand the transport of free fatty acid from the adipose tissues to the target cells. As we just consider the human body which possesses the fats which have adipose cells in them. As fats are present in different sites of human body like abdomen, thighs and arms. Adipose cells consists of triglycerides that possess glycerol in combo with three free fatty acids. The aggregation of glycerol with three fatty acids is important because glycerol consists of a large molecular mass that check the escape of free fatty acids from these adipose cells. An enzyme lipase performs its function when breaks the bond between the glycerol and the fatty acids. Once its bond is degenerated, the free fatty acid can pass into the bloodstream and actually reach any cell of the body that is able to metabolize free fatty acids. Two tissues are also located in the body that cannot metabolize free fatty acids, which are the RBCs and the nervous tissues. Both of these tissues possess only one thing in common, that they have no mitochondria, and these mitochondria are essential for fatty acid metabolism. Students, this is a target cell that can metabolize the free fatty acids with the help of mitochondria. The free fatty acids must enter into the cytoplasm of the target cell by the special protein known as fatty acid transporter. These free fatty acids have a net negative charge and they cannot pass the membranes of the cells, so they require a special transporter to pass through the cell membranes. During the next step, the free fatty acids must pass into the mitochondrial matrix after passing its outer and inner mitochondrial membrane. Inner mitochondrial matrix is essential because it contains all the enzymes that are needed for the beta oxidation of fatty acid, like the enzymes for the Krebs cycle and the electron transport chain. Before the free fatty acids can be translocated into the mitochondrial matrix, it must be activated. Next, we study about the activation and transfer of free fatty acid. Therefore, first we see the fatty acid activation. A free fatty acid must be converted into its active configuration, acyl CoA, because acyl CoA is the only type which can be metabolized in the mitochondrial matrix. This reaction is catalyzed by the enzyme acyl CoA synthetase, and during this reaction an ATP molecule is changed into monophosphate and a pyrophosphate molecule is also generated and then released. Coenzyme A is also needed for this reaction, as this reaction happens at various points in the cell, including the outer mitochondrial membrane, the endoplasmic reticulum, and the peroxisomes. The fatty acid activation is very important because without the fatty acid activation it cannot be utilized as such in the mitochondria. So when once activated, the free fatty acid are then taken into the mitochondria. Now we understand this transfer of fatty acid in a slight explanation. Suppose at this side we have the cytoplasm of a cell. We possess the outer mitochondrial membrane here and the inner mitochondrial membrane here. The space between them is known as intermembrane space. At this side we possess the mitochondrial matrix. The outer mitochondrial membrane contains these little pore-like structures known as porins that prepare the outer mitochondrial membrane sufficiently permeable to different materials, including acyl CoA. So, acyl CoA can pass through the intermembrane space after passing through the outer mitochondrial membrane. On the other hand, the inner mitochondrial membrane is non-permeable to materials like acyl CoA. So we need a special mechanism to pass the acyl CoA like materials across the inner mitochondrial membrane into the matrix. This mechanism is called the Carnitin shuttle mechanism. This Carnitin shuttle comprises many enzymes, and one of them is the Carnitin palmitoyl transferase 1 enzyme located in the outer mitochondrial membrane. This carnitine palmitoyl transferase 1 enzyme performs a function that it attaches a carnitine molecule to the acyl coenzyme A. In this reaction, the coenzyme A of acyl CoA is released back into the cytoplasm and a molecule of acyl carnitine is generated inside the intermembrane space, is produced inside the intermembrane space. During the next step, the enzyme carnitine, acyl carnitine transferase that is present in the inner mitochondrial membrane, take this carnitine into the mitochondrial matrix. But as we just talked about, acyl CoA is the only form of the fatty acids that can be metabolized inside the mitochondrial matrix. so it must be degenerated. This function is performed by the enzyme carnitine palmitoyl transferase second. This enzyme attaches coenzyme A again to the acyl molecule and regenerates the carnitine back. The carnitine that is resynthesized back is transferred again into the intermembrane space that can be used by the carnitine palmitoyl transferase 1 enzyme. Students, you can understand from this lecture that the enzyme carnitine acyl carnitine translocase shift one molecule of carnitine into the intermembrane space, and in conversion of in exchange of that it carry one molecule of acyl carnitine into the mitochondrial matrix. So this is the whole mechanism of that how the free fatty acid is transferred into the mitochondrial matrix. Till now we have learned the transport of free fatty acids from the adipose tissues to the target cells. We have also discussed about the entry of free fatty acids into the cytoplasm and the mitochondria. Next we articulate about the oxidative catabolism in detail that occurs inside the mitochondrial matrix. So for the complete understanding of the beta oxidation topic, please watch this video till end, because now we will conversation about the oxidative catabolism of fatty acids that takes place in the mitochondrial matrix. So now let's start with what happens inside the mitochondrial matrix, that is the prominent part of the beta oxidation. Students, this is the basic formula of a fatty acid to which a side chain is attached to a carboxyl functional group. If we detach a hydrogen atom from this compound and attach a coenzyme A group to this compound, this becomes a fatty acyl CoA. The word fatty in the name of fatty acyl CoA can be removed depending upon the length of the side chain that is associated to the functional group. For example, a side chain with 16 carbons associated, the fatty acid will be called as a Palmitoyl CoA, and a fatty acid with a side chain of 14 carbons is Maristoyl CoA, and 12 carbons will be called laurel CoA. When this fatty acyl CoA comes into the mitochondrial matrix by the carnitine shuttle mechanism, it is acted upon by four enzymes whose main function is to separate two carbon atoms from the parent fatty acid. Now these reactions lead to the generation of two molecules. The first one is the acetyl coenzyme A and the second is a new fatty acyl CoA, which is two carbons inferior than the original parent fatty acid, that in this case will be 14 carbons compound Maristoyl CoA. Now the main point about this beta oxidation pathway is that the newly formed fatty acyl coenzyme A, that is two carbons inferior than the parent compound, reintroduces this pathway. Therefore, we start with a 14 carbon compound Maristoyl CoA, which is acted upon by the same four enzymes, and this leads to the production of a new fatty acid, that is a 12 carbons compound fatty acid. This also leads to the production of a new molecule of acyl CoA. With every cycle of beta oxidation, the fatty acid side chain becomes shorter. Palmitoyl CoA, when truly metabolized by these four enzymes, then provides 8 molecules of acetyl CoA. Now let's consider how a 16 carbons fatty acid, the palmitoyl CoA, is thoroughly degraded into 8 acetyl CoA molecules by the beta oxidation process into mitochondrial matrix. Therefore, we start this example with the 16 carbon fatty acid, palmitoyl CoA. Here you can watch the alpha and the beta carbons of this fatty acid. Here the first reaction which happens during the series of beta oxidation is a dehydrogenation reaction which leads to the displace of hydrogen. Students, hydrogen atoms are deleted from the beta and the alpha carbons, which causes the formation of double bond between them. The compound that is obtained is called Transenoyl CoA, and the enzyme that catalyzes this reaction is called acyl CoA dehydrogenase. Students, you can analyze clearly that the hydrogen atoms are taken out from the alpha and the beta carbons, and these hydrogens are then transferred to the electron acceptor, which in this case will be the FADine, that is reduced to FADH2. So the resultant molecule is known as transenoyl CoA because the two hydrogen atoms are now in trans state. The second reaction which happens in the series of beta oxidation is the hydration reaction, which means addition of water molecule. This water is added around the alpha and the beta carbons. A hydroxyl group is added to the beta carbon and a hydrogen atom is added to the alpha carbon, and the compound obtained is known as hydroxyacyl CoA, and the involved enzyme is known as enoyl CoA hydratase. The third reaction happening is again a dehydrogenation reaction. A hydrogen is also displaced from the beta carbon of the hydroxyacyl CoA, which leads to the formation of a double bond between carbon and oxygen atom. The hydrogen that is separated is accepted by the NADH, which is reduced to NADH, and this reaction is catalyzed by the enzyme hydroxyacyl CoA dehydrogenase, and the compound obtained is known as ketoacyl CoA. The ketoacyl CoA is a much more symmetrical and balanced molecule which can be degraded into two molecules. So the last happening reaction is the cleavage reaction called thiolases. This reaction also utilizes a coenzyme A molecule, and it is catalyzed by the enzyme thiolase. The ketoacyl CoA is then split into two molecules. The first one is a new fatty acid which consists of about a side chain of 14 carbons called Maristoyl CoA, and the second molecule is the acetyl CoA. Students, deeply consider this reaction and try to analyze that how the ketoacyl CoA is degraded into these two molecules, because the first three reactions that happen during the beta oxidation series just prepare the fatty acid for acyl CoA to a more balanced and symmetrical molecule that can be split into two molecules. The prepared Maristoyl CoA again passes to the beta oxidation series, and it is thoroughly utilized to form 8 molecules of acetyl CoA through the similar reactions. Next, we derive the summary reaction and the energy produced from the beta oxidation. Up till now, we have delivered a lecture how a 16 carbon palmitoyl CoA is converted into 8 molecules of acetyl CoA. From these reactions, we have also known that reducing equivalents are obtained, that are the FADH2 and the NADH2. Students, for a palmitoyl CoA, which is a 16 carbon fatty acid, to be entirely degraded into 8 acetyl CoA molecules, it has to undergo 7 beta oxidation cycles. During these 7 cycles of beta oxidation, a 4 carbon side chain is obtained, which is entirely degraded into 2 molecules of acetyl CoA. So after these 7 beta oxidation cycles, we attain the following compounds, which are 8 molecules of acetyl CoA, 7 molecules of NADH, and also 7 molecules of FADH2. All these procedures occur in the mitochondria. So we possess the enzymes for Krebs cycle and electron transport chain in a close proximity, where all of these substrates can be used for the totaling of for the estimation of energy. 8 molecules of acetyl CoA are taken to the Krebs cycle. The 7 molecules of NADH and also 7 molecules of FADH2 are taken to the electron transport chain for the generation of energy. Next, for the estimation of How many ATPs are generated during the beta oxidation? You must know 1 acetyl CoA molecule obtains 10 ATPs, passing the Krebs cycle. So make sure to watch the video on Krebs cycle for better acknowledgement of and consideration of beta oxidation. As one molecule of NADH is equivalent to 3 ATPs and one FADH molecule is equivalent to 2 ATP molecules. As I have already described during the video of total energy calculation from one glucose molecule during electron transport chain, but this is really the situation because not all the time all the substrates are utilized in the electron transport chain because a minimum amount of energy is also wasted during the movement and in carrier proteins. So for the energy estimation we take one molecule of NADH as 2.5 ATPs and one FADH molecule is considered to be equivalent to 1.5 ATPs. So estimating the energy obtained from beta oxidation of palmitoyl CoA, 8 molecules of acetyl CoA will provide 80 ATPs from the Krebs cycle. 7 molecules of NADH will provide 17.5 ATPs from ETC and 7 molecules of FADH2 will provide 10.5 ATPs from the ETC. So in totaling we possess 108 ATPs. Students, from this all energy calculation, it is crucial to reveal two ATPs that are utilized during the activation of Palmitoyl CoA. So the energy obtained from 16 carbon fatty acid is 106 ATPs per molecule of Palmitoyl CoA. So students, it is our topic beta oxidation today. If you are liking it, then press the like button and subscribe our channel for receiving the notifications of further upcoming videos. At the end, I want to encourage you that you have a debt of your country and you can pay back it with getting education with hard working, bona fides and sincerity. It is the only thing, students, it is the only one thing that you can do in your present time for your country, because students, you are the future of your country. So please get the education with hard working and sincerity. Thank you for watching this your video.